Hello, everyone. Welcome to today's live broadcast, Unpacking USP 922 and Water Activity's Role in the Pharmaceutical Industry. I'm Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be moderating today's event. We're pleased to bring in this webcast presented by Pharmaceutical Technology and sponsored by Meter Group. I'd like to share a statement from our sponsor. Meter Group produces a suite of water activity measurement and analysis instruments for the pharmaceutical industry. The company recently introduced the Aqualab TDL, the only water activity meter capable of accurate and reliable measurements on highly volatile compounds. From cleansing radioactive Fukushima rice paddies to helping discover water on Mars, Meter Group has helped companies in a wide range of industries solve complex moisture-related problems. I've got a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your question by typing it in the Q&A box, which is located on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, just click on the question mark Help widget in the dock at the bottom of your presentation window. Now, before I welcome today's speaker, I'd like to invite our audience to participate in a brief polling question. And you can make your selection directly on the screen and click Submit. So here's the question. Do you or your company currently use water activity measurements? Yes, regularly in several functions or departments. Yes, regularly in one department. Yes, but only rarely. Or no, not at all. Again, the question is, do you or your company currently use water activity measurements? Yes, regularly in several functions or departments. Yes, regularly in one department. Yes, but only rarely. Or no, not at all. So make your selection on the screen and click Submit. And thank you for participating in our first poll. Now I'm pleased to introduce today's speakers. Dr. Tony Cundell is a member of the current USP Microbiology Committee of Experts, the joint committee that developed USP 922 water activity. He takes a leadership role in the area of modern microbiology methods. He consults with pharmaceutical, consumer health and dietary supplement companies, microbiology instrument manufacturers, contract testing labs, and sterile compounding pharmacies in the areas of microbial risk assessment, regulatory affairs, and microbial testing. He's also co-chair of the PDA task force responsible for the 2014 technical report number 67, Exclusion of Objectionable Microorganisms from Non-Sterile Drug Products. So thank you for joining us today, Dr. Cundell. I'm going to turn the presentation over to you.
Thanks very much, Rita Peters, for that introduction. Yes, my name is Tony Cundell, and my topic today is the USP role in the promotion of water activity determination in the pharmaceutical industry. First, a few disclaimers. I am an independent consulting microbiologist to the pharmaceutical industry. I'm not an analytical chemist, so my emphasis will be on microbiology today. I am a member, as Rita said, of the 2020-25 USP Microbiology Expert Committee. My presentation today is my opinions and not necessarily those of the U.S. Pharmacopoeia or my industrial clients. So this is an overview of my presentation. I'll make a brief introduction to water activity. I'll discuss the concept of hurdle technology as applied to drug products. I'll discuss the role of water activity in mitigating the risk of microbial contaminations in non-sterile drug products, and I'll discuss the activities of the USP committee in this particular area. First, a little whimsy. This is the planet Earth, which is a wet planet, and because of the water on Earth, our planet teems with life. In contrast, Mars is considered a dry planet, so it's inhospitable to life. And yes, this is a photograph of the actor Matt Damon, who appeared in the science fiction movie The Martian. So what is water activity? Water activity, or the equilibrium relative humidity, measures the vapor pressure generated by the moisture present in a hygroscopic product. Water activity equals the ratio of P divided by P0, where P is the partial pressure of the water vapor at the surface of the product, and P sub 0 is the partial pressure of water vapor above pure water at a specified temperature. So water activity reflects the active part of the moisture content, or the part which can be exchanged between the product and the environment. Water activity is a unitless ratio, and it ranges from 1.0 for pure water to 0.0 for a bone-dry material. So what are some of the applications of water activity determination in the pharmaceutical industry? It can determine the level of thermodynamic control for a desired solid form. So for the production of a hydrate versus an anhydrate. It can be used for selecting excipients, where the water activity may in part impact their material flow, compression characteristics, hardness, and dissolution of a solid dosage form. You can use water activity in optimizing fluidized bed drying processes. You can reduce the degradation of active ingredients within product formulations by controlling the water. And this is especially important to those active ingredients that are susceptible to chemical hydrolysis. Also, water activity can be used to establish the level of protection to product formulations to moisture by the primary packaging material during the shelf life. And it can be used as a complementary method to the Karl-Fischer titration for monitoring changes in water content. So if you construct a water absorption isotherm and show the relationship between the Karl-Fischer titration for water content and the water activity, you can do the simpler measurement of water activity to determine the water content. So in the area of microbiology, you can optimize formulations to improve the antimicrobial effectiveness of preservative systems. By controlling water activity, you can reduce the susceptibility of formulations to microbial contamination. You can use water activity as a stability test parameter to confirm that the packaging protects a drug product from moisture and that maintains the microbiological stability of the product. And you use water activity as a tool to justify the reduction of microbial testing of different non-sterile formulations. So next I'll discuss hurdle technology. You may ask, what is hurdle technology? Well, it's a concept that was developed in the food industry, and it can be applied to drug products. So hurdle technology deliberately combines existing and new preservation techniques to establish a series of preservative factors, so-called hurdles, that the microorganisms in question are unable to overcome, like jump over. And these hurdles may be storage temperature, water activity, acidity and alkalinity, redox potential, antimicrobial preservatives. And at the bottom of the slide, I have the reference to the pioneering publication on hurdle technology for food products. Next, I want to discuss microbial risk analysis for drug substances and drug products. So the question you ask when you do a risk assessment is, what is the water activity of the material? Is water used in the final steps of chemical analysis and isolation of the drug substance or active pharmaceutical ingredients? Does the manufacturing process increase, reduce, or eliminate bioburden? Does the manufacturing environment and process equipment contribute to microbial contamination? And does the manufacturing site have a good history of microbial control? And similarly with drug products, a key question is the water activity of the product. Also, you would consider the microbial quality of the pharmaceutical excipients, the manufacturing process, the environment, the testing history, the packaging, whether it protects the product, whether it's a multi or single-use product, and what is the medical status of the recipient of the product. So next I want to discuss the role of water activity in the mitigation of the risk of microbial contamination in non-sterile drug products. So water activity has been extensively used in the food industry to prepare products with a low risk of microbial contamination and also to extend the shelf life of the products. And this use of water activity goes back to 1000 BC in China, where drying, smoking, salting, and spicing was used for food preservation. So water activity can be successfully employed in the cosmetic, consumer products, and pharmaceutical industries, as well as the food industry. So here's some of my favorite low water activity foods: pickles, sausages, smoked salmon, and jams. So where did the linkage between water and activity and microbial growth come from? Well, the Australian microbiologist Bill Scott in the early 50s showed that microorganisms have a limiting water activity level below which they will not grow. Water activity and not water content determines the lower limit of available water for microbial growth. And here at the bottom of the slide is the references to his pioneering papers. So what generalizations can we make about water activity and microbial growth? So all microorganisms have a minimum and optimal water activity requirement for growth. Drug products with water activity below this requirement will be self-preserving or microbiologically stable. And these findings can be used in microbial risk assessment and the establishment of microbial specifications and testing programs. So this particular slide shows the water activity requirements for different types of organisms. So at high water activities above 0.95, bacteria outcompete yeast and molds. Below 0.85, most bacteria do not grow. Below 0.75, most yeast and molds don't grow. Between 0.75 and 0.60, only highly specialized microorganisms not usually found in pharmaceutical products will grow. This could be microorganisms in the Dead Sea. And below 0.6, no microorganism will grow. So this gives it a lot of opportunity to control microbial growth. Here is the water activity requirements of a range of well-known microorganisms. On the left is bacteria, on the right, molds and yeast. As I said, gram-negative organisms have the highest requirement for water activity. Gram-positive organisms, a lower requirement. And Staphylococcus aureus does not grow below 0.86. And the same pattern is seen with yeast and molds. There is a small group of highly specialized organisms, halophilic bacteria, osmophilic yeast, and xerophilic fungi that will grow at water activities approaching 0.6, but they grow very slowly. So this lends itself to a division between wet and dry products. So a high water activity above 0.8 represents a moist system, and a low water activity below, say, 0.7 represents a dry system. And you should be aware, if you expose a high water activity product to a low humidity, you will lose water, and conversely, if you expose a low water activity to a high humidity, the product will gain water. So this gives a division between low and high risk products based on water activity. So in the low risk, we have things like ophthalmic ointments, topical ointments, liquid-filled capsules, liquid-scented cough drops, powder-filled capsules, compressed tablets, suppositories, and inhalation aerosols. High-risk products, these would include nasal sprays, inhalation solutions, rectal creams, liquid antacids, eye drops, oral liquids, topical lotions, cough syrups, oral canker sore gels, and oral analgesic suspensions. So quite conveniently, products segregate out into a low risk or low water activity products and high risk and high water activity products. So next I want to discuss the USP involvement in the area of water activity, especially chapters 11, 12, and 922. Just a little background. The U.S. Pharmacopoeia has test chapters numbered between 1 and 999, and they contain general test chapters that may be cited in an ingredient or a product drug monograph. So in other words, if your monograph for a particular material has a specification, it will have a counterpart with a test that's described as a general test chapter. In contrast, USP chapters numbered between 1,000 and 1,999 general information chapters that provide broad guidance but are not binding. These chapters are not GMPs. GMPs are derived from the government regulations and FDA guidance documents. So first I want to discuss the development of 1112. I can take some credit here. Bob Friedel and myself co-authored a stimuli article that was published in the Pharmaceutical Forum in 1998, and it was entitled "The Application of Water Activity Measurement to the Microbial Attributes Testing of Non-Sterile Over-the-Counter Drug Products." And yeah, I was reminded that this is many years ago. So the USP published a general information chapter in the Pharmaceutical Forum in November, December of 2002, and this was entitled 1112, Application of Water Activity Determinations to Non-Sterile Pharmaceutical Products. And the chapter became official in the second supplement to US 29 in August of 2006. So it's been in the USP for some time. However, with the development of USP 922, water activity, the Microbiology Expert Committee is revising 1112 to focus on the microbiology aspects of the chapter. So what's the difference between the two chapters? So 1112 is a general information chapter, provides guidance. It focuses on risk assessment for microbial growth, describes basic calibration of instruments and salt solution. At the time when we wrote this chapter, we found the dew point chilled mirror instrumentation had been published as an official method in the AOAC International, so we referenced that particular method. It does not discuss a wide range of instrumentation, and it does define water activity and how it can be applied to non-sterile drug products and microbial testing. In contrast, 922 is a general test chapter, and it's focused on water activity test methods for all types of drug products, contains recommendations and requirements for instrument qualification, calibration, and measurement, and it describes the types of instruments that can be used and their pros and cons, and lists their possible use. And it defines water activity and includes additional information about how the measurement works and what factors affect the result of the measurement. So you can see the difference in intent of the two chapters. Again, this is some of the applications of water activity, which I discussed earlier. So what was the evolution of this particular chapter? It was first published in the Pharmaceutical Forum in November, December of 2018 as an in-process revision with stakeholder comments. And based on public comments that were received, it was rewritten, and we significantly improved the chapter based on the comments. and it was published in the Pharmacopoeia Forum in January-February 2020. So the proposed new chapters outlines the recommended concrete methods to qualify, calibrate, and use water activity meters to accurately measure water activity in raw materials and products. It is a little unusual compared to other general test methods because it discusses water activity broadly and recommends a number of measurement technologies, not just one. So this is the table of contents of the chapter. It has an introduction, theoretical background, factors affecting water activity, the application of water activity measurements, types of water activity instruments, qualification of these meters, operation and calibration of the meters, procedures for verification and validation, and a section on the operation, calibration, and validation of near-IR instruments, and it has 3 references. So the 4 major types of instruments discussed are the dew point temperature chilled mirror instrument and 2 types of electronic hygrometers and the tunable diode laser technology. And it does discuss the near-IR spectrophotometry. I won't go into details of these technologies. As I said, I'm merely a microbiologist, not an analytical chemist. But I will say that the user should select the instrument based on their use. So you have to consider the level of accuracy and precision that you would require. And typically, microbiologists are interested in a lower level of accuracy and precision, where chemists favor a high level of accuracy and precision. You consider the throughput of samples, whether the measurement is affected by some attributes of products like containing volatile materials, whether you want to take a sample or do a measurement within inside a container. So you have to consider what use you're going to put the instrument to and make a decision what would be the best instrument for your particular lab. So one area that was not discussed in 1112 was sample preparation. So 922 emphasizes that during transportation from the lab, samples need to be kept in well-sealed, water-impermeable containers to protect them from any change in water composition due to exposure to different temperature and humidity in the sampling site in the lab. And also, it's recommended that sample container have a minimal headspace to avoid significant changes in water activity due to re-equilibration of the sample to the environment in the sample container. Also, it discussed the sample measurement, and calibration checks or performance checks should be performed every day the instrument is used to perform measurements. And also, you should verify the instrument is not due for its periodic calibration prior to performing a measurement. So you record the equilibrium water activity and the equilibrium temperature and report the value or the mean value of replicate measurements. Typically, you report to 2 or more significant digits depending on your application. And you report the temperature rounded to the nearest 0.1 degree because water activity does change slightly with temperature of measurement. The sample presentation may be important. The user will tell you how much material should be in the sample chamber. And also you have some options in terms of sample preparation on how the sample is presented to the instrument. For example, a compressed tablet could be presented as whole units or a ground tablet. Chewable gels could be sliced into thin pieces, and capsules could be presented as whole units or the capsule content. And based on these presentations, you may see slight differences in the water activity measurement. Instrument classification. The USP has an excellent chapter, 1058, Analytical Instrument Qualification. And it categorizes instruments, lab instruments, into Group A, B, and C. Group A, which I don't describe here, trivial devices that are used in the lab, like mixers or stirrers. Group B instruments, a typical example is a pH meter or an oven. And these group only require routine calibration, maintenance, and a performance check. And the extent of these activities may depend on the criticality of the application. And also, these instruments may have firmware, but not software that is updated or changed by the user. Group C instruments comprise of analytical instruments with a greater complexity and significant degree of computerization, such as high-pressure liquid chromatographs and mass spectrometers. And all elements of qualification, including software validation, must be considered to ensure the proper functioning of instruments in this group. But, the take-home message is most of these meters fit into the Group B. So you can use USP 1058 as a guidance document. Yes, I've basically covered this slide. So in contrast to Group B instruments, there may be more requirements with near-hour instruments. And this is discussed in USP 856. At this time, I'll go back to Rita. We have our second polling question.
Well, thank you, Dr. Cundell. Yes, we have our second poll question. So once again, audience members, I invite you to participate. You can make your selection directly on the screen. So the question is, which water activity applications are you most interested in? Is it hurdle technology and/or preventing microbial issues? Ensuring chemical or API stability? Influencing compaction, hardness, and/or dissolution rates? Shelf life studies and/or extending product shelf life? Maximizing excipient effectiveness? Or is it other applications? So once again, the question, Which water activity applications are you most interested in? Hurdle technology and/or preventing microbial issues? Ensuring chemical or API stability? Influencing compaction, hardness, and/or dissolution rates? Shelf life studies and/or extending product shelf life? Maximizing excipient effectiveness? Or other applications? So if you just make your selection and click Submit. And thank you for participating in this poll. And now we'll move back to the presentation.
Thanks very much, Rita. So calibration checks or performance checks, which are typically done each day you use the instrument. So saturated salt solutions may be prepared in a sample cup or prepared in a separate container and transferred to a sample cup for use. And you would use these material according to the recommendations of the instrument manufacturer in regard to the amount of sample that would be present in the measurement chamber. So in addition to the saturated salt solutions listed in 922 Table 1, purified water may be used as a 1.00 water activity standard solution, but it's not required to include a standard for zero water activity, and it's acceptable to use an appropriate low water activity standard such as lithium bromide or lithium chloride. So the procedure for standard solutions are the instrument should be calibrated either using a prepared or purchased solution or even using a calibrated humidity generator. For prepared solutions, you should reference Table 1. And when do you calibrate? You would do that at least annually. Any time that the calibration check or performance check fails, and any time you change the range of intended use. And you would calibrate as instructed by the manufacturer. You would calibrate at the temperature of use where it's feasible. You would bracket the sample, or if you have a large range, you may also have a midpoint sample. So in terms of performance checks, it's a bit like certainly to an unsophisticated microbiologist, like a pH meter. Here are the saturated salt solutions, and they range from high water activity to low water activity. And in the right-hand column, there is the value and the precision you would expect for measurements at 25 degrees. So I did mention that there are references in the chapter, and I was honored that the book that I edited with Anthony Fontana, Water Activity Applications of the Pharmaceutical Industry, were included, plus two excellent standard references to water activity measurements. I just want to take a minute to give a shout out for people who are involved in stability programs. Water activity is an excellent tool to measure the protection of the product by the primary packaging material. I was involved, this is a war story. The company I work for marketed an oral contraceptive in a blister pack in a South Indian nation, and this particular product failed. The woman taking the oral contraceptive became pregnant. And what we found was the blister packaging by material differs in the moisture transmission. And the transmission rate was high enough in a very high humid area for the water to penetrate the packaging and accelerate the chemical hydrolysis and degradation of the active ingredient. So this is a great tool. So what is the USP plans in the area of water activity? Stakeholders' comments were on the revised chapter 922 have been received and reviewed. And if you work for a company that made submissions to the USP, that certainly made our work easier and resulted in a much better chapter. So where appropriate, additional revisions were made to Chapter 922. The chapter was approved by ballot by the Physical Assay Expert Committee, and I'm pleased to announce that it will be published as official in May 2021, in a couple of months. And in response to this publication, the USP Microbiology Expert Committee will remove the theoretical background and details of analytical methods from 1112 and cite the Chapter 922 and focus on microbial risk analysis. So, in conclusion, I believe the USP has provided an important analytical tool with water activity measurement to help develop better non-sterile drug products. I will thank my colleague Martin Coffey from Bosch & Lomb for the use of a couple of slides from a future presentation, and I'll take questions as time allows. Thanks very much for your attention, and I give it back to you, Rita.
Well, thank you very much, Dr. Cundell, for that informative presentation. Before we get started on the Q&A, just a quick reminder to our audience members that they can submit questions by typing them in the Q&A box, which is located on the right-hand side of the presentation window. So here's our first question. So how is water activity used to help support skip lot testing for non-sterile products?
To be frank, I'm not a big fan of skip lot testing. In my experience working in product development, I would recommend that you do a risk assessment and decide what risk of microbial contamination your product has. So a compressed tablet, for example, has a very low risk of microbial contamination. And write up your risk assessment, and in the submission to the FDA, suggest that a microbial specification is not required, so then it does not become a specification. And if you have a controlled process, you control your water system, your ingredients, your manufacturing process, you should consistently get a low water activity, low bioburden product. And skip lot testing would not be necessary. That's my answer to that question.
All right. Well, thank you very much. Here's the next question. So what are your thoughts on using water activity measurements to replace Carl Fisher titrations?
Well, as a microbiologist, colleagues have complained about the smells that might come out of incubators in the microbiology lab. The Carl Fisher measurement uses pyridine, which is an unpleasant solvent, and it's a relatively complicated assay. So water activity measurements could be substituted for water content measurements. But I would even go further. In many cases, the critical parameter is not water content, it's water activity, and maybe you should file with a specification for water activity for that particular product and not water content. The only downside is you have to construct water absorption isotherms for each product so that you can use water activity to derive a water content measurement. I hope chemists listening today believe that I've got it right. Next question.
Sure. Here it is. So if water activity data is very low, say 0.6, can we justify removing the micro-testing?
Yes. If the product is protected by the packaging material, it won't change during the shelf life. And water activity will be a conservative property. So if you use the same formulation and manufacturing process, you should consistently get a product that will have a low water activity. And no organism will grow at that particular level. So the product will have microbial stability. So yes, I think you can make that decision.
Thank you. Here's the next one. What kind of water migration from capsules to powder should be expected in lower moisture environments? And what would be a comfortable range to observe?
Yes, I did mention water will migrate from a product to the environment depending on the relative humidity and water activity of the product. So I'm not an expert on moisture migration, but obviously the primary packaging material will protect the product during the shelf life. And if the pharmacist dispenses the capsule, they usually only give you a 30-day supply, and the packaging that they use will continue to protect the product. So I really haven't answered the question. So my feeling is the emphasis should be on the product protection.
All right, thank you. Here's the next question. What's the difference between water activity and moisture content, and when should each be used?
Well, it's an energy situation, and the water content could be chemically locked up in the product, but still measurable. But the water activity measures water that is free water. I know water activity experts don't like it to be described as free water, but it's water that can migrate from the product to the environment. And in lots of cases, that is the driver for the parameters that you're interested in, whether it will be flow of a powder, chemical degradation of an active ingredient, or sufficient water to support the growth of organisms. So I would suggest that in many cases, in formulation development, the formulators and the chemist should determine whether it's water content or water activity which is going to be the critical parameter, and make a decision on what one to use. Next question.
All right, sure. So of the major types of water activity instruments, is there one type you found to be most reliable and or valuable?
I find there's tremendous merit in all of them. I have to admit, when I got started in this particular area, I did use the dew point chilled mirror instrumentation, and I did find it was robust. But there is some of these electronic devices are much more suitable for in-process control or testing powdered material. So I'm going to punt on this one and say you need to evaluate how you're going to use these instruments and make a decision on which one you would use. In my experience, they are all quite reliable, but there is differences in accuracy and precision and how they use, and also cost. So you need to decide which one would be best for your purpose. Next question.
Well, actually, at this point, Dr. Cundell, we're going to wrap up. If we didn't get to anyone's question, it will be answered offline. I want to thank the audience for attending and participating in today's event, and I'd like to thank our sponsor, Meter Group, for making today's webcast possible. Our audience members will receive an email alerting them when this webcast will be available for replay, and I invite you to forward that announcement to your colleagues who may have missed today's live event. So thank you again for joining us, and have a great day.
And thanks very much for the opportunity of talking on this particular topic. Thank you, Rita.